also an associate professor in Earth and Environmental Sciences at the CUNY Graduate Center. And he is with Brooklyn College. Brett got his doctoral degree in oceanography at the University of Connecticut, and then did a postdoctoral fellowship research at the in Western Australia. He has now returned back to Brooklyn, where he studies urban waters of Brooklyn and its surroundings. He has a number of additional affiliations. He is also the New York co-chair of the New York, New Jersey Harbor and Estuary Program. He is the president of the New England Estuarine Research Society. And actually, incidentally, I keep getting emails. There is a conference scheduled this upcoming spring. So I want to give everybody a little ping for that one, a little bit of advertising for you. And he also sits on the board of the Coastal and Estuarine Research Federation. And he's, generally a great person to get to know. And today he's going to speak with us today about the water quality of Jamaica Bay. And I'm very excited to hear the seminar. So with further ado, I hand it over to Dr. Branco. Thank you very much.
Thank you so much, Diane. Let me just get my presentation up on the screen and we'll launch right into it. Great. So thanks for inviting me. I'm really excited to speak with you all. On the menu for today, I want to talk about Jamaica Bay, in particular, the sort of quest for meeting the Clean Water Act goals of fishable and swimmable waters, and the great challenge that is. And I'm kind of hoping that what this does is inspire perhaps research. This is an area where there's lots of different disciplines that can make contributions. And in my role as the director of the Science Resilience Institute at Jamaica Bay, I'm always trying to inspire coalitions of researchers to get together and work on things of interest to Jamaica Bay, but that are also applicable to other sort of similar urban systems. So as Diane said in the intro, right, I do have these affiliations, and I wanted to highlight them because one of the things that all of these affiliations have in common is that they all kind of operate at that boundary between research, policy, and society, right? So for example, the New England Estuarine Research Society, an awful lot of that membership are people from different agencies across New England, some of them with more of a research focus, but a lot of them are practitioners, mixing in with sort of academic researchers. And there's always like these really rich exchanges, between, all of these different, stakeholders, if you will. The same could be said for the Coastal and Estuarine Research Federation. And it's really interesting to sit on the Harbor and Estuary Program Water Quality Working Group as an academic, because most of the members of that group are, from, for example, Department of Environmental Protection, city parks. There's a bunch of people from New Jersey, the the Harbor Discharges Group. There's just an NGOs. So it's really interesting to sit on there. And actually, as an academic, it's really important because you get to hear what the real needs are of those different communities. And I've always been a big fan of trying to align my research, with the policy and stewardship needs, of the communities that I'm embedded in. So I try as much as possible to to work in that space, and so you'll see a little bit of that in my presentation today. I just wanted to highlight, I am currently serving as the director of the institute, and the institute is all about supporting planning and decision making for climate adaptation and environmental stewardship in Jamaica Bay. And we try to engage in a variety of things: research, education, and a lot of public engagement or community engagement, or however you want to characterize that. And before we sort of officially started today, I was just telling Professor Pakar how it's one of my goals to get more and more CUNY faculty involved in the work of the institute. And so maybe this might spur some follow-up conversations about that. So let's get right into our our topic. So, so we're talking about water quality today. And, one of the first things that, everyone's always wanting to know about water quality. How's the water in Jamaica Bay? Is it safe? You hear people ask that kind of question a lot. Or even, the waters of the harbor, are they safe? Can you swim in it? Is it safe to touch? But water quality is really, in some ways, it can be a bit of a nebulous concept, right? Because it's context dependent. So if you're talking about water quality, are you talking about water quality with respect to fishable waters? And by fishable, that means, water bodies that can support fisheries, right? That's good habitat for fish that are commercially important and the food webs that they depend on. But we can also think about water quality in the of organisms and the suitability of that water as habitat. So, for example, is the water quality such that it can support oysters, or is the water quality, to the point that it can support eelgrass beds or some other important sort of marine habitat? So water quality is a bit of a, it is very context dependent. And so when we talk about, how's the water quality, we oftentimes need to be specific about the context in which we are discussing it. One of the challenges of water quality in places like New York City and the harbor and Jamaica Bay is just some of the history of the watersheds that surround that bodies, right? The water quality of the water bodies is intimately tied to what's happening in the watershed. And if we think about places like Jamaica Bay, so in this old map from 1844, Jamaica Bay is this landmass, part of this open bay, in the southern part. And from this old map, you can kind of get the idea that, first of all, this bay was not developed, right? The fringing land is mostly salt marsh and highly dynamic sandbars and sand dunes and sand spits, which means that it's very dynamic and constantly changing. You can kind of see in this old map too that a lot of the land of Brooklyn and Queens was starting to get turned over to agriculture. So you can kind of see all the different farming plots in here. But then if we look, today, 150 years later, what we see is all of the, and I'm calling them irreversible changes. And I think that that's a fundamental fact about urban watersheds that that limit our, the things that we can do to impact water quality, right? So we now have, a very high population density. Most of an awful lot of the marshland that used to fringe Jamaica Bay has been covered with a thin layer of sand and a thin layer of pavement and neighborhoods. The bay has been dredged. At one time there were plans to turn Jamaica Bay into, the shipping center for New York City and therefore the world. And they actually did a lot of dredging to deepen the bay, straighten out a lot of the old marsh creeks to make them places that they could moor ships. But then that actually never happened. And then you can also see the imprint of, I hope you can see my cursor moving. I'm kind of hovering around what is now Floyd Bennett Field, which was New York City's first major airport. And then over in the eastern part of the bay, you can see the footprint of JFK International Airport, which was built not that long after Floyd Bennett Field and sort of displaced its role as a commercial airport. So there's been lots of irreversible changes in Jamaica Bay, which means that that you can't necessarily turn back the arrow. The arrow of time only goes in one direction. So when we think about things we can do to improve water quality, we have to think about transformations that move us forward, not not trying to take us back to some previous time. So I'm going to talk about water quality in the context of the Clean Water Act and what the state actually mandates by law, the water quality of Jamaica Bay and also a lot of the other waters around New York City. But the state has legal classifications of the water quality for water bodies based on their intended use. So, for example, most of the waters of Jamaica Bay are classified as SB waters, which means that their primary use is primary and secondary contact recreation and fishing. That's the goal, which is why I called this talk about the goal of attaining swimmable and fishable waters, right? And these waters shall be suitable for fish, shellfish, and wildlife propagation and survival, right? So not only does it speak to the fishable, swimmable, but it also kind of pays homage to the to the ecological, to the habitat. And most of the tributaries of Jamaica Bay, which used to be marsh creeks, but have since been straightened, deepened, and bulkheaded. But anyways, those tributaries are Class I waters, meaning primary use of secondary contact recreation and fishing. Again, suitable for fish, shellfish, and wildlife propagation and survival. I like the last little clause there, though. Other factors may limit the use for this purpose. And again, I just want to emphasize that this is state law. And so, the DEC, the state's Department of Environment and Conservation, is really in charge of making sure that we move towards, attaining those uses of the water body. And, those are sort of like narrative descriptions of the use, but we can start to put some specific criteria to these sort of fishable and swimmable goals. And I'm just showing you a subset because I'm really going to talk primarily about ultimately dissolved oxygen and indicators of human sewage in the waterways. So I'm going to focus on sort of DO, total coliform, and fecal coliform. But you can see that there's attempts to put specific goals for these parameters on Class SB and Class I waters, and right. So for example, the dissolved oxygen of these water bodies are allowed to be between 3 and 4.8 for short periods of time, but ultimately the goal is for these water bodies to be above 4.8 milligrams of oxygen per liter, and that's mostly based on its use as habitat by fish and the amount of oxygen that they need to thrive. The interesting thing about Class SB and waters is that this 3 milligrams per liter is one of those thou shalt not fall below 3 milligrams per liter at any time. And we'll talk a lot about that in a bit. But those are goals, right? So when we talk about the quest for fishable, swimmable waters, one of those quests is to have a water body in which dissolved oxygen does not fall below 3 milligrams per liter. And then we also have, criteria for indicators of human raw sewage, total coliform, fecal coliform. Now, this is an area which I have less expertise, so I will talk less about it, but I will certainly mention it because the quest for dissolved oxygen and the strategies to attain these goals is often part of the same strategy. So let's talk about what is the root cause of water quality problems in a place like New York City. I think that you could make an argument that water quality in urban estuaries is primarily a food problem and our infatuation with bacon cheeseburgers. And it looks delicious to me, maybe not to some, but the reason this is important is because when you take like the Jamaica Bay watershed, right? The landmass surrounding Jamaica Bay, which wastewater is collected from and then eventually directed towards the bay, has somewhere around 2 million people. And those 2 million people all eat, and most of what they eat, they then later, becomes waste, human waste. And that food, of course, is not created in the city, right? That food is brought in from outside the city. So there is a net import of the stuff that makes up food. I'm going to highlight nitrogen here because nitrogen is often seen as one of the primary drivers of the degradation of urban water bodies, at least for marine water bodies. So, so you can make estimates of how much, for example, nitrogen that you import into the city, usually mostly in the form of food, which then gets concentrated in in the places that we dispose of our waste. In this case, places like Jamaica Bay, right? So the waste of 2 million people goes to Jamaica Bay is the bottom line here. So if we take, if we just take a rough estimate of how much nitrogen per person per year, and then look at, the roughly 1.6 to 2 million people that are in the watershed, then it's easy to see, it's about 9, gigograms of nitrogen per year that comes into the Jamaica Bay watershed or sewer shed, and then subsequently gets returned to the waterways. And this creates all kinds of problems. So that's why I say, in large part, water quality is a food problem. Also, right, like I mentioned before, a lot of that food that we eat gets turned into waste, and then of course that gets into our sewer systems and eventually gets discharged, maybe after treatment, into the bay, and that waste also carries, pathogens. And so not only is food, part of our nitrogen problem, but it's also ultimately, I think, it's food plus the concentration of people that's the root cause of our pathogen problem in the waters around Jamaica Bay. And it's a huge problem, right? So if we look at the amount of nitrogen, so this is the nitrogen load to Jamaica Bay in kilograms per day, pre-development, and this was all done part of a USGS study back in 2006. So pre-development, they estimated that, in the absence of colonial settlement, there was about 35.6 kilograms per day, and that's just sort of natural, right? Nitrogen fixation goes through the ecosystem and decays, and then, gets into the bay through groundwater or surface runoff. And we can look at the order of magnitude change in the amount of nitrogen loading to Jamaica Bay from 35.6 to, this was 2006 numbers, but somewhere around 15,785 kilograms per day, the vast majority of which was coming through the wastewater treatment plants, meaning it's a food problem. But look at even the groundwater is an order of magnitude higher than it was pre-development. So it's a big problem to overcome. Back in the 70s, we were fortunate enough to, to have the passing of the Clean Water Act, and that might singularly be one of the most important, events to alter the trajectory of water quality in water bodies around the United States, right? Because the Clean Water Act, which basically is what called for fishable, swimmable waters in the United States, resulted in, over time, billions and billions of dollars in investments in water pollution control plants that would treat waste, disinfect it, take some of the gross stuff out, get rid of some of the carbon, which was causing big problems in our waterways. There were stricter rules about discharging heavy metals and organic pollutants like PCBs and DDTs into our water bodies. And this paper from O'Shea and Brosnan in 2000 kind of summarized a lot of the positive benefits that the Clean Water Act had, up to 2000, not just in Jamaica Bay, but also in all of the waters around New York City. So I do want to kind of talk a little bit about, in a minute, I'll talk a little bit about some of those specific improvements that we have seen. But basically, the Clean Water Act, resulted in, one technological solution, which was the construction of lots and lots and lots of wastewater treatment plants or water pollution control plants, or however you want to label them. It's really interesting to just remember or think that, it wasn't until the early 20th century that we did anything at all to treat raw human sewage flowing into Jamaica Bay. And the Coney Island wastewater treatment plant was one of the first in the country to be designed and built. But some parts of the city didn't have their wastewater treatment plants until well into the 20th century. I think like in Gowanus, the Red Hook plant. The Red Hook plant, I think, dates back to the 1970s or something like that. So, and there's still a lot of investment in wastewater treatment plants, primarily again to reduce the amount of human pathogens and to reduce the amount of carbon in our waste that makes it into our water bodies. And these investments, again, you can see a lot of these investments were made in New York City. Here, like I was saying, the Coney Island 26th Ward and Jamaica plants in Jamaica Bay were some of the first in the country to be built around the turn of the 20th century. That construction continued all the way through the 60s and the 70s, and then now in the 70s through the 90s, these plants are getting old, and new technology was developed, and so they went through extensive upgrades. But fast forwarding into the last 20 years, last 15 or 20 years, one of the technological advances in wastewater treatment has been nitrogen removal technology, right? Because the excessive addition of nitrogen into our coastal waters has stimulated primary production to the point where you start to have lots of adverse impacts, including reduced oxygen levels, harmful algal blooms, and things like that fundamentally alter the habitat and pose new human health risks. So the city actually invested pretty significantly and still is investing pretty significantly in technologies to try to reduce the amount of nitrogen that's being discharged into our local waters, through upgrades to these plants. So let's take a brief look at some of the improvements, right? Because this is not all a bad news story, right? There's lots of good news about water quality in New York City, for sure. So for example, since the mid 80s, there have been significant, if you look at it from like a summer mean amount of human pathogens that you find in Jamaica Bay, there's been a significant reduction, and now we oftentimes see values, not always, but frequently the values for these pathogens fall below what's considered safe, by New York State's standards. However, and I'm going to see if this link works. So this is we'll see if this works. Are you guys now seeing a web page, or are you still seeing my PowerPoint?
We're still seeing the PowerPoint.
Okay. I'm going to stop sharing for a second then, because I think this is important enough to share with you. I'm going to stop sharing and share my internet now. Okay, so in spite of those reductions, I want to show you these data from a citizens water quality testing program run by the New York City Water Trails Association. There's a network, a citywide network that for quite a few years now spend their summers every Thursday. Networks fan out across the city and take a water sample, oftentimes near some sort of public recreation access point. And then they deliver those samples all around the same time to labs around the city. And actually, Queens College is one place that these samples have been delivered to. I think that's Greg O'Mullen's lab. We at Brooklyn College have helped with this, but they also have labs at NGOs around the city, and therefore testing for some of those indicators of raw sewage. And when you look at these data, the story is not quite as good. So for example, I'm just going to quickly show, hopefully you're seeing this. Are you now seeing a spreadsheet?
Yes.
Yes.
Right. So, so the bottom line here is that through this citizen science testing program, red means their samples were higher than the New York City Department of Health standard for bathing beaches. Green means it's okay, and yellow means close to the limit, basically. And if you just kind of move across this table, you'll see lots of red. And then if we move down to places like the Newtown Creek, and I'm scrolling down, here we go. So Coney Island, here we go, Jamaica Bay, Pathogen Basin. These samples are collected by a group called the Sebago Canoe Club, who like to paddle around Jamaica Bay. And if you just kind of scroll across their line, you can see that they're very rarely below the state limit for safe waters. So even though it's definitely in some ways a good news story, right? In some ways it's a good news story because the general trend is decreasing pathogens. We still have a ways to go to make the waters swimmable for New York City. So let's talk about some other aspects of water quality, and I'm showing you this map to indicate places where the New York City Department of Environmental Protection has routinely been collecting water samples weekly in the summer, monthly in the fall, winter, and early spring, to look at standard indicators of water quality like dissolved oxygen and chlorophyll and nutrients and water transparency, et cetera. And I'm going to probably mention these stations J1, J11, and 9, in comparing to a lot of these stations in the interior. So if we look at dissolved oxygen, again, these are sort of like average summer values, at individual stations. And what you can see is station J1 was right there at the mouth of Jamaica Bay, and generally speaking, it's always had pretty good water quality, right? Dissolved oxygen values that are up somewhere around between 80 and 100%, which means it's typically well above even 4.8 milligrams per liter. But as you move from the inlets sort of like deeper and deeper into the bay, like so for example, from into station J5 and then J3, which is near Canarsie, and J7, which is way in the far eastern reaches of the bay, you see changes. And I want to focus on this J7, the Bergen Basin. Bergen Basin is a basin which has a wastewater treatment plant outfall, and so it's one of the most impacted places in the bay with respect to the discharge of wastewater. And you can see that the water, the dissolved oxygen levels in general are lower near the bottom of the water. So DOB means bottom and DOT means top. So it's like measuring near the bottom of the water column and near the surface. The general trend at J3 and J7 has been improving dissolved oxygen concentrations at the surface of the water bodies, right? There's been some improvements in dissolved oxygen, but if you look at J7, like over this period from the 1950s into like the mid 90s, there's lots of variability near the bottom, but in general, there's no real clear trend of improving dissolved oxygen conditions, in spite of all those wastewater treatment plant improvements, over that time period. So a little bit of a mixed story if you look at specific locations around the bay. We look at something like chlorophyll A, which is an indicator of the the amount of production in the bay, which is which we oftentimes presume is driven by excessive nutrients. And you can see that in the early 80s, it was relatively low, and there was a big increase into the into the mid 90s. And that, that had to do with a change in the way that they were operating the wastewater treatment plants. And they actually ended up discharging more nutrient-rich water into the bay in the mid 90s. But since the mid 90s, there's been significant reduction in the amount of nitrogen, which I'll show you in a minute, being discharged into the bay. And in response, it looks like, right, that chlorophyll has been steadily declining since the mid 90s in response to a lot of that improvement in the technology. And you know, and if we look at it in terms of the amount of nitrogen that is actually being discharged into the bay from the wastewater treatment plants, you know, the New York City Department of Environmental Protection has had a goal of trying to reduce discharges by 50% from their mid 90s levels. So, you know, in the mid 90s, they were discharging around 50,000 pounds per day, and they're trying to get down here, you know, between 20 and 30,000, and they've been getting there. They've been getting there because of the upgrades that they have been doing to the wastewater treatment plants. However, I want to draw your attention back to one of the earlier slides when I showed you that the nitrogen discharge in the bay is many magnitudes higher than it was pre-development. So a reduction of 50%, while admirable, does not get us anywhere near what pre-development nitrogen loadings were to Jamaica Bay, right? And so we shouldn't expect water quality to return to the condition it was, you know, back in the 1700s or the 1600s before, you know, before we started to extensively develop the watersheds around here. There's a big difference between cutting something by 50% versus cutting something by 2 orders of magnitude, is my point. So a former master's student of mine had done a little bit of work using the Department of Environmental Protection's data, trying to tease out some cause and effect relationships between management actions and changes in water quality, and also trying to tease out some relationships between different parameters. So so one neat thing she did with the time series was to look for breakpoints in the trajectory of water quality, right? So rather than just like putting a straight line through the whole time series to look at trends, what she was doing is she was looking for significant changes in the trends themselves. So she did this breakpoint analysis, and the cool thing is, like for total killed-all nitrogen, which is organic nitrogen plus ammonia, so it's the biggest pool of nitrogen in Jamaica Bay. She clearly detected across all of the stations in Jamaica Bay a significant change point that occurred around 2009 in Jamaica Bay. And in the 3 stations, J11, N9, and N16, those are the ones that are just outside Jamaica Bay, that change point occurred in 2010. But it was really consistent across all the stations, which was really cool because you can kind of tie that to some of the changes that the DP was making to the way they operated their wastewater treatment plants. The interesting thing, though, is, you know, one of the paradigms for enrichment of marine water systems like Jamaica Bay is that productivity is driven by excess of nitrogen. So you would expect to see, you know, maybe you could hypothesize that you would see synchronous changes in the response of these parameters. But interestingly enough, what Danielle found was that there were significant changes in the trends in chlorophyll, but they preceded by, you know, by 4 to 5 years, the significant changes that she saw in the trend of total killed-all nitrogen, which again is most of the nitrogen in the bay itself. So that's a little bit of a that's an interesting finding, which just sort of points out that that these systems don't necessarily behave as cleanly and neatly as we might expect them to. I'm going to actually skip the next 2 slides just because I want to make sure that I finish in plenty of time to have questions. Another thing that that Danielle did was she did a principal component analysis to try to tease out relationships between different important water quality variables. And this one was kind of interesting, right? Because I know you can't read the labels, but I'll just walk you through it a little bit. This cluster of arrows to the right are the nutrients and chlorophyll, and actually water temperature, curiously enough. The arrows that are pointing almost directly down are pH and dissolved oxygen. And I'm highlighting dissolved oxygen because that is one of the, remember, that's one of our goals, our water quality goals, is to improve dissolved oxygen, because that's what the state says we have to do. And the interesting thing about this is that in this sort of principal component analysis, the fact that the dissolved oxygen is almost orthogonal to the nutrient and chlorophyll axes suggests that that the relationship isn't quite as, or actually that there isn't a strong relationship between those. So what that tells me is that, and you can kind of see it in the time series data, is that all of these wastewater treatment plants that reduce nitrogen have not had a significant impact on dissolved oxygen. We're not seeing the payoff.
Brett, you just
Yes.
we just lost you for about 20 seconds.
Okay. How is it now?
You're fine now.
Okay. I was probably saying something nonsensical, so I think I'll just forge ahead. Basically, I was just, actually the point I made was that the investments in nitrogen reduction have not yet really manifested themselves in improvements in dissolved oxygen concentrations, particularly near the bottom of the water column, which is an important thing for policy and management, right? And we'll talk a little bit about that in a moment. Just to reiterate the points about being able to see these, these changes in the trajectory of chlorophyll and nitrogen. Remember, I said that there was a breakpoint a couple of slides ago, right? We saw this breakpoint in chlorophyll around 2006, 2005, 2006, and you can kind of see it in the time series here, right? Prior to 2006, there was a significant decline in chlorophyll. Over the last 7 years or so, there's been no significant change in chlorophyll, even though that's when we've been seeing big reductions in the amount of nitrogen in the bay. So again, these things that sometimes we expect to be related aren't always, the picture is a lot less clear, which presents management challenges, right? And there's also a lot of limitations to using data sets like those that are generated by the DEP, because when you take samples once a week at a specific time of day in a limited set of locations, you lose a lot of the detail about the dynamics of these water quality parameters. So a few years ago, Chris Gobler and Ryan Wallace from Stony Brook had put out some in situ dissolved oxygen sensors so they could continuously monitor dissolved oxygen concentrations in the eastern part of the day. And the main story here is that if you look at this particular location that they were monitoring, if you went out in the middle of the day, which is when most of these most of this time series peaks, you might measure and see very healthy water quality concentrations. But note that an awful lot of the time, the water quality, the dissolved oxygen is less than 4 and even 3 milligrams of oxygen per liter, and that primarily occurs at night, or it might occur for a longer period of time. But if it doesn't happen when the DEP is out there doing their sampling, it'll be completely lost. And so there's a mismatch between trying to attain this dissolved oxygen goal and the way that we monitor for it, quite frankly. So let's get to so some of the things that we need to do to help drive us closer towards trying to attain these fishable, swimmable waters goals. So, we talked about wastewater treatment plant upgrades and, reducing carbon and reducing pathogens and nowadays reducing lots of nitrogen. But we still have a problem that that the city's stormwater and wastewater systems are so antiquated that during rain events, we oftentimes have to discharge raw sewage directly into our water bodies and subsequently expose people to all of those pathogens. So the DEP around the city has been investing heavily in so for example, storage facilities. So this is a combined sewer overflow control station at the head of Pathogen Basin in Jamaica Bay. And what happens is when it rains, instead of releasing that raw sewage out through the combined sewer overflows, it gets stored in these there's a 50 million gallon storage facility, basically subsurface beneath this facility that fills up when it rains with that water. And then when the storm event passes, that water is then pumped back up and out and to the wastewater treatment plant so that it can be treated and sent out to the bay in a better state than it would have otherwise. So the city is employing technologies like this, or strategies like this in other parts as well. There's only so many places that you can put 50 million gallon tanks in New York City, and so there's some limitations to this approach to trying to improve water quality. There's been significant investments in green infrastructure, right, to intercept stormwater, do some treatment, let nature do the treating before sending that water on its way to places like Jamaica Bay. But again, there's only so much area in New York City where you can build green infrastructure, where you can put the green infrastructure. Right now, the city is looking at schools because the schools are one of the biggest landowners in New York City, and it's sort of like the last frontier for major green infrastructure projects. And then there's there's other approaches like constructed wetlands. We're restoring a lot of wetlands in the city, not just for the habitat benefits, but also because we think that that it can enhance nitrogen removal from the waters of Jamaica Bay, and perhaps even contribute to improved dissolved oxygen conditions. And the DEP has even been designing wetlands to try to maximize their potential to remove pathogens from combined sewer overflows. So they've got this one that they built up off of Valley Creek up in your neck of the woods by Queens College. And we're actually trying to get some work with, I think, with Diane Greenfield and Greg O'Mullen and I are trying to, get a contract done with the DP so that we can assess the performance of this constructed wetland and its potential for nutrient and pathogen removal. So, so there's potential for using nature or nature-based solutions to try to help get us further towards those water quality goals. And just a couple of more slides. I wanted to sort of bring this back up now to big picture for a moment, maybe to kind of summarize, right? So, so we have this wonderful policy tool called the Clean Water Act, and we have all these wonderful technological solutions that we've been coming up with and applying over the years, all trying to move us closer to fishable, swimmable waters. But in parallel with that, we've been gaining we've had an evolving understanding of the relationship between things like, for example, nutrient loading, nitrogen loading, and the response of the water body, like the dissolved oxygen levels and the chlorophyll and system metabolism, et cetera. And Jim Clarn out of San Francisco was the lead author on this great paper in 2001, which kind of outlines how our understanding has evolved from this very simple model of we load the system, it responds, to something that has evolved to look more like this, right? It's not just nutrient enrichment, which is the top box, but there are multiple stressors on these ecosystems like Jamaica Bay, climate change, overfishing, toxins, introduction of invasive species, aquaculture, manipulations of the watershed hydrology. And all of these stressors are filtered through the unique physical and ecological, structure of the water body to affect some perhaps very complicated responses that then create human health costs or benefits. There's social and economic implications. Lots of impacts which drive our will to invest in infrastructure and more science, frankly, to figure out how to rehabilitate, and I hate the word restoration, but transform these systems and their stressors to provide more positive responses, right? Because we want the coastal ecosystem to benefit us, not to pose harms to us. And we also understand, so this gets back to one of my original points about the arrow of time. And Carlos Duarte and some others in 2009 wrote this wonderful paper called Return to Neverland. And for them, Neverland was the way things used to be. And the main thesis of this paper was that, it's naive to think that we can simply reverse, the impacts of increasing nutrients over the last couple of centuries. You just reverse that and expect the ecosystem to return to the way it was, because in that time, baselines might have been shifted. Thinking back to the previous slide, all these other things were happening too. Climate was changing, and humans were affecting fish populations and food web structures, etc. So the baseline shifted. Like what we can return to has actually shifted. And not only that, but we're starting to understand more and more that a lot of these ecosystems exhibit complex behavior. And once you shift it from one state to another, it might be that that new state is fairly stable, and you might have to reduce the insult, the nutrient inputs, below a certain level before you would even expect to start to see a response. In other words, it's not a perfectly linear forwards and backwards situation. And I would posit that perhaps what we see in Jamaica Bay is we just haven't reduced nitrogen, for example, below some certain threshold to affect the kind of responses that we wish to see. And then, of course, you can have regime shifts on top of shifting baselines to make for some very complex, ecosystem response to things like nutrient inputs. So the last couple of slides were really just to say that trying to attain fishable, swimmable waters is not such a simple task. There's a lot more we have to learn and understand about how these systems respond to all the different stressors that I showed you a couple of slides ago. And so the work never ends, right? And there's still, some people say we've been studying the nitrogen problem for 20 or 30 years, and we have. But we probably are going to be studying it for another 20 or 30 before we really, perhaps, right, before we really understand what's going on. And with that, I will stop. That's my main message for today, and I hope you enjoyed this little tale that I just spun for you.
Well, thank you. That was a great talk, and I loved it. So I bet there's going to be some questions from some of the audience, including some of the students. So you can either use your microphone or you can just type it in, whichever one you would like to do. Okay, it'll take a couple seconds for typing, because most people are shy. Okay, so we actually have a question from Robert to start it off. Decades ago, New York City made freshwater impoundments and ponds in Jamaica Bay because people at the time had an idea that fresh water is better than brackish water. So by impounding fresh water, they were improving the bay. We know that's wrong now. You've mentioned constructed wetlands. Do you have any other concerns about them transforming the environment and doing so? Nice question.
Yeah, That's a super interesting question, and I don't think we can talk about freshwater being quote unquote better, but I think I want to respond to that by telling a quick, simple story. And one of the places where they created some freshwater impoundments in wetlands is the Jamaica Bay Wildlife Refuge, and maybe that's specifically what you're referencing in that question. And the West Pond, I'm sorry, the East Pond, no, the West Pond. The West Pond in the Jamaica Bay Wildlife Refuge was created by building an impoundment, right? An earthen dam. And that was breached during Hurricane Sandy. And so that formerly freshwater pond, which by the way, was initially filled with New York City tap water, which is a whole nother story. But that freshwater pond basically became a tidal marsh system. And the changes in the surrounding vegetation were almost immediate, right? And you could start to see the transformations going around there. So the National Park Service had to go through a long process of figuring out what to do, if anything. And so they went through a long process, but turns out one of the loudest constituencies and strongest constituencies arguing for rebuilding the impoundment and turning that West Pond back into a freshwater impoundment was the birding community in the Audubon, the Audubon societies, because they see that that freshwater habitat as being critical for maintaining some of the bird diversity and some of the species diversity in the bay. So I would say to that question, I don't think it's there's lots of different ways to look at at the freshwater versus brackish water impoundment and the intent of creating those. I don't think it's quite as clear cut of a story maybe as that question implies, but I also think it's up to the part of the work of the Science Resilience Institute at Jamaica Bay should be doing the research that can help inform those kinds of decisions, knowing that those decisions are going to be made not just on the scientific information, but also on what the communities in the most holistic sense want to see happen. I hope that's a responsive answer to your question, Robert. Should I just go through these questions as I see them?
Yes, so let's go on to the next question from Tim. Interesting apparent correlation similarity between dissolved oxygen and pH on the slide from Chris Gobler's data. Any ideas why that is?
Yeah, and Tim, I was probably, when I showed the PCA analysis showing that the DO and the pH weren't correlated with the nutrients, the DO and the pH are certainly strongly correlated with each other, and I think that's to be expected, right? Because even though it is a marine system, and even though it's those pH changes are somewhat buffered, those day-night fluctuations in dissolved oxygen and CO2 are so large that I just think that you're seeing the DO and the pH changes sort of synchronous. I've seen this. I used to do a lot of work in shallow, not used to, I still do a lot of work in shallow lakes where the water system isn't buffered, where the pH isn't buffered, and you see the same thing, just more extreme fluctuations in the pH.
Okay, so I have another question from Max this time. You mentioned baseline shifts being fairly permanent. Wondering if this is the case with the low oxygen levels at depth. Does the data suggest that the surface oxygen levels respond to WWT, but that there are other factors acting on longer timescales impacting the bottom?
Yeah, that's a great question as well. And I think that there's a lot of things. I think oftentimes we use too simple of a mental model about oxygen and how it's going to respond. So, for example, we know that the bay is deeper, right, because of all the dredging in the borrow pits. than it was a long time ago. And some of those deep parts are more strongly stratified than they've ever been. And so, you know, it's a simple, so in some places in the bay, I think you might not ever really be able to impact those low DO levels because you just can't transport oxygen down there fast enough to keep up with, say, like maybe the sediment oxygen demand. So that's one. I think another thing is that another thing would be you know, climate change and the impact that has on water temperatures. So we know that the dissolved oxygen, right, saturation is going to be impacted by water temperature, but also some of the climate effects might be to more strongly stratify water systems like Jamaica Bay, either through you know, warmer surface waters or more freshwater runoff if we get more precipitation in certain areas, right? So I guess what I'm really trying to say, Max, is it's a good question because I think that you know, we need better reaction and transport models for dissolved oxygen to be able to better predict how it's going to respond to things like reduced nitrogen loading.
Okay. So, oh, excellent. And so we have another question from Aslan. What exactly is a hot moment? And what is the significance of nitrogen fixation, particularly in the ability between anammox and denitrification to occur?
Yeah, hot moment. So there was another paper that came out. I can't remember the year, and I'm sure some of the geochemists probably saw that as well. A paper about hot spots and hot moments. And the general point of the paper was when we do things like monitoring programs where we collect samples in specific locations at some preset time interval, oftentimes we miss the events where, so for example, where maybe biogeochemical rates of transformation are at a high point, right? Like maybe after a rainfall event or, you know, some other driver that might cause, you know, a couple of days of really high you know, respiration rates or nitrogen denitrification rates or something like that. Or it might even be particular places. You know, we sample a bunch of places around Jamaica Bay and try to assume that we know something about average rates of things like nitrogen cycling. But if we're not sampling those hot spots for reactions, we could be way off in our estimates. So hot moment really is just referring to those times when biogeochemical rates might be accentuated, but we're not measuring them, or maybe we are. We wish we could. Does that answer your question?
Okay. And so we're starting to run out of time, but we have a couple more questions. You have a few more minutes, Brett?
Sure.
Okay, great. So this is from Godfrey. What analytical techniques can be used to differentiate between, oh no, we already did that one, sorry. No, actually, no, it is new. Okay, Godfrey. What analytical techniques can be used to differentiate between anammox reactions when they occur at the same time with DRNA reactions?
Well, first of all, Godfrey, how are you? I think it's been a long time.
I'm doing well. I know it's a long time.
Yeah. Yeah. You know what? Do you mind? Can I kick that question over to? I hate to do this, but I don't know if Diane has a better answer. I hesitate to answer that because I think that I don't know the answer off the top of my head because I haven't made these measurements myself.
Well, you can do PCR reactions for the active genes in both of them. So Godfrey, you took my biogeochemistry course, right? I know you took one of them.
Yes, last semester.
Yeah, so you know that they're similar reactions, but they function fundamentally differently. So there are actually PCR reactions that can identify specifically which ones they are. And anammox in particular there are genes that you can specifically identify and look at their activity.
Okay,
Excellent.
thank you.
Okay, thanks.
Thank you.
You know,
Thanks,
so
Diane.
this
And
is
I
so-
have to say real quick that my wife and daughters would be so proud that I just said I don't know to a question.
well, you know, Brett, you know, what I do in my class is that I tell the students if I can't answer a question to something that I'm teaching, they can email me later in the evening that question and I'll give them extra credit.
Right.
Because then I want to Google that. I want to find it out. So, so we stopped the professor today. Congratulations,
Yeah.
Gottfried. Absolutely. So, so anyway.
So, Steve, may I have a few questions?
Oh yeah, sure.
Do you have time? Yeah. So, Brian, thank you. In your talk in the last two slides, there's a concept. You summarize with the concept model. So, because your institute is a resilient center, so this use resilient concept to organize your data. So for the resilient parameter, you looks like use chlorophyll A or PPP, right? That's primary production. Yeah. So control parameters, maybe oxygen level or nitrogen level. Yeah. My question is, do you have the PPP data?
So you mean actual measurements of primary production?
Yeah.
Yeah, no, that's a great question. That's actually one of the so those data do exist for like shorter term studies. So for example, when Chris Gobler did his work a few years ago, he was making production measurements of the phytoplankton, but also of macroalgae, which I didn't even mention in the bay, which is something that the DEP doesn't even capture in their survey program. And I know that for the past couple of years, the DEP has been doing sort of like an enhanced data collection program where they have been trying to get measurements of primary production and respiration, et cetera. But they just don't exist sort of like as a continuous time series like some of these other parameters.
So another question is, you show your J7, that's the data. There looks like a significant gradient of nutrition level between top and the bottom. What the fundamental reason?
For the vertical gradients?
Yeah.
Yeah, well, I think that well, first of all, like it's not quite like a shallow lake, but you know, high rates of reaction, some stratification. I mean, there is some stratification. You know, there's some significant inputs of fresh water through wastewater treatment plants that can help to sort of stratify the water column and set up some of those vertical gradients. They're typically stronger in the eastern part of the bay than the western part of the bay. So, you know, and then you also get some significant thermal stratification, especially, you know, more like a diurnal pattern in the thermal stratification. But, you know, I just think it's just, you know, in a highly productive ecosystem like Jamaica Bay, you don't need much stratification to start to set up some really strong vertical gradients. Does that answer your question?
Yes, that looks like a physical reason, not a chemical reason, right?
Yeah, it's physical.
Oh yeah, absolutely. Yeah.
Okay, thank you very much, Brad.
Okay, so I have one more question, if you can hold on, from Bill. Okay, and so how has the relatively high variability observed in the high resolution data impacted confidence of drawing conclusions from the apparent trends seen in low density data sets?
That's a great question. I kind of started to allude to that a little bit, right? When I showed the high resolution dissolved oxygen and pH data. Yeah, no, that's actually a really important question. I think that the low resolution data, the low density data sets can tell us something about 20, 30-year trends. So they are very useful in that sense. However, you know, in terms of you know, trying to attain management goals, or even trying to fundamentally understand how the system is working, I think that those low density data sets are very inadequate and lend low confidence in some of our sort of like cause effect relationships and understanding of system response. I hope that's an okay answer.
Yeah. Okay. Well, it is 1:23, and I think that we're probably wearing you out at this point. But so I'd like to ask everyone to give a virtual applause again. That was a great talk. We really enjoyed it. I enjoyed it. And so thank you so much.
Thank you. Thank you. Thank you very much for having me. And hopefully I'll hear from some of you soon if you want to get involved in some work